Hello, my name is Caleb, and this presentation is over biofilms. I'm going to throw a couple basic definitions at you really quickly so we know what we're talking about unilaterally. A biofilm is a complex congregation of microorganisms that is held together by a matrix that adheres and protects the complex. We'll talk extensively about this matrix. It is called an EPS, or an extracellular polymeric substances. Whew, man. That is also in the complex with bacterial cells as well as proteins and DNA of the various cells, as well as things that constitute the EPS. And then now we're going to go over the creation of these biofilms, and these are 4 steps. The initial being initial attachment. This first step includes surface adhesion using the pili, the fimbriae, or the flagella. Once attaching, metabolic processes soon follow that leads to the formation of a microcolony, which is the second step. These microcolonies, or I like to think of them as a lag phase if you're just thinking bacterially, that is the establishment of that population within that surface, and they are proliferating their DNA as well as getting everything correct to go into the maturation or the log phase, which is the development of the biofilm, the EPS complex, and other complex architecture. After the log phase is hit, the reason why they call it log phase is bacteria grows in logarithms, meaning 2 logs and then the logarithm, and it is a logarithmic reproduction cycle. But as well as that, with the logarithmic growth and the increase drastically of the population density, we'll touch more on the significance of that later, but it has to do with the signaling, which we'll touch on once again, like I said, a little bit later. After the maturation is dispersion. Now, some bacteria that may not be synergistic to the complex and consequently detach. So that goes towards that population density. If it's a false population density, they're going to fall off because you trim the fat in biology. And all of this population density, this EPS, the communication, all towards one thing, and that is increased survivability. Why does anything in nature come together? It's because they are symbiotic or they're synergistic. In this case, they are synergistic. Some things are symbiotic until a synergistic wave moves towards that biofilm. Like I said, the survivability increases due to the synergistic effects of the population density. Now, this is for a multitude of things. This is because the EPS matrix, which we've already touched on briefly, this protects microbes from disinfecting agents as well as antimicrobial drugs through the prevention of penetration into the complex and killing those cells directly. And when I say microbial drugs, I'm also talking about the immune response, which also we will go over later. Another reason for the increase in the survivability of the biofilm is because of the genetic exchange horizontally to share other antimicrobial resistant qualities. We'll talk more on that when we go over the population density, but that also attributes to that signaling aspect of the gene expression, which is a huge reason why we'll go into the quorum sensing of the biofilm. Now, this is cell-to-cell communication. This ties in all of the increased survivability. That's why I've kind of been dancing around the subject, because I have a lot of things I need to tie into this one thing. The quorum sensing, once again, is the cell-to-cell communication. The cells used to coordinate gene expression modifications. Now, whenever this population density rises to a proper threshold, the cells that each just innately excrete these autoinducer signalers just innately. So there's already a congregation just based on the density. It's like a marker. It's a here in attendance, if you will. You call your name here. So the here inducers, the here signalers build with population density, which we just said, which in that logarithmic stage, the population density obviously skyrockets based on the replication rate logarithmically increasing as compared to additively increasing. Once this threshold is rates these changes in gene expression occur. The impact of this is it regulates EPS shielding, as well as increases that the gene expression for the antimicrobial properties that protect against, obviously, antimicrobial treatments, disinfectants, human immune system. Those survivability characteristics are shared through those gene expressions, which we'll get into with the interactions with the immune system. The immune evasion is done through the matrix because it hides pathogens from phagocytosis and other immune mechanisms, as well as therapeutic intervention, which we've already touched on extensively. That EPS is a key shield to all of the pathogens that are hiding within it. Obviously, within it there is antimicrobial resistance that is shared through that horizontally sharing of genes. So that is the inner protection, but the ultimate protector of these biofilms is that EPS. So the evolution and the more complex of the EPS, the more protected and more resistant these biofilms and therefore pathogens are. So that is the base evasion, is it can't attack in the first place, so it has no idea it's there until obviously the pathogenic effects already take place. 2, the chronic infections. Once you get these biofilms, it's hard to get away. Now, the biofilms cause infections and they don't go away because of their resistance to both the immune system and therapeutic modalities. And there's a few ways to remove these, and I would like to take a few moments just to talk from experience because this personally impacts my everyday career. I work in orthopedics as a physician extender, and we throw in obviously arthroplasties like crazy, and the big thing that we talk about is plaque on teeth, because after we replace someone's knee or shoulder or whatever joint, you pick it, and once we put a prosthesis in there and we go in for a teeth cleaning, the biofilm on the plaques of teeth get into the bloodstream, which bloodstream goes to the knee and at an accelerated rate because the inflammation, obviously a higher metabolism rate in that area. Obviously the bacteria wants to go to a high metabolism rate, which is obviously a healing area within the body, which creates inflammation, biomarkers, all that good stuff, all that good biochemistry, microbiology stuff going on inside of that prosthetic joint. And that attachment happens, obviously the proliferation. But anyway, we'll get to the brass tacks of why I brought this up, is to get rid of these biofilms. Me personally, I know our protocol is we like to up the coverage of the antibiotics, because obviously you got gram-positive, gram-negative, you got 100 different things you're fighting against inside that biofilm, which is another huge contributor to that survivability, is because you can't kill them all with one thing, because you're fighting a losing war against things that you're already having a difficult time. Now that they're synergistic, they're sharing their worst qualities that we're trying to fight with each other. So not only are we upping the coverage of our antibiotics to cover a multitude of things, we like Doxy, Keflex a lot of the time, but the ultimate solver is physical removal. And for us, that means the removal of a prosthesis for 6 weeks with an antibiotic spacer to obviously try to break up those biofilms. We put a multitude of antibiotics as well as quorum sensing disruptors. So that is our next way to treat these biofilms, is to disrupt their EPS, or disrupt them from disrupt them from forming in the first place. But since that is not an option in our scenario right now, we want to break up the EPS so that coverage in those IV antibiotics that we have someone on, we have a PICC line in for 6 weeks and a spacer on the bone that has been infected by these biofilms and these pathogens for 6 weeks. Now during that time, this is a pretty extensive protocol because of how resistant they are, so we have to get a multitude of coverage as well as those quorum sensor disruptors, lowering that EPS and that survivability to hopefully get in and clear everything before an 8-week period passes, and we pull the PICC line out and then we put a new knee in. But there is a huge recidivism rate in arthroplastic infections because of these biofilms not clearing all the way, regardless of our best efforts. But anyway, so this has been my tangent on biofilms. If you have any questions related to my specific scenario or anything regarding biofilms, let me know. So, thank you. Bye.